right, so today what we have left to cover in chapter 11 is a little bit on synapses. So this is basically going from one neuron into another neuron. That's a synapse. So we're going to talk about that, and we're also going to be talking about drugs and drug addiction today. So notice I told you a synapse is from one neuron to another neuron, but it can also be from one neuron to an effector. And remember, an effector can be any of your muscles. Ooh. Okay, so it can be skeletal muscle, cardiac, or smooth muscle. It can also be your glands. Okay, so glands can also be effectors, like sweat glands. So the nerve comes in, right? It looks very similar to the neuromuscular junction, right? Whether we're talking about a nerve going into another neuron, or whether we're talking about a neuron going into a muscle or a sweat gland or some other effector. That's always what it looks like, that sort of synaptic cleft and that junction in there. It always looks like that. So we say that when we're going from one neuron to another neuron, we have a presynaptic neuron. This is what's going to be conducting the impulse towards the synapse. So in the drawing that I did up here, here's our presynaptic neuron. And then we have our postsynaptic neuron. And this is going to be transmitting the impulse away from the synapse. So this will be the second neuron or the effector. Okay, so this one down here would be the postsynaptic neuron. That should make sense if we think of these prefixes, right? Pre means before. And post just means after. So the presynaptic neuron is before that synaptic cleft. Postsynaptic neuron is after the synaptic cleft. Now, the types of synapses that we can have, if we're talking about neuronal synapses from one neuron to another, we can have axodendritic. This means that the axon of the presynaptic neuron, okay, so let's draw our neuron out, here's our axon, okay. The axon of that presynaptic neuron is going to connect to the dendrite of the postsynaptic neuron. Okay, so it would look something like that, going from the axon into a dendrite. We can also have axosomatic. So the axon of the presynaptic neuron is going into the soma or the cell body of the postsynaptic neuron. Or we can have axoaxonic, where the axon of the presynaptic neuron is actually connecting to the axon of a postsynaptic neuron. Okay, so those are our three different kinds. And here's a great picture from your book that shows you these. So you can see here, remember I told you that yellow in your textbook is always going to be a neuron. So if we look here, we've got this postsynaptic neuron that's really big in yellow. And we can see our presynaptic neurons are all coming in here. And they're all different colors. So here we can see the axon coming in and connecting here to the soma. So this would be axosomatic. Here we can see the axon coming in and connecting to the dendrites. So that's axodendritic. And here's the axon coming in, connecting to the axon. So that's axoaxonic. Okay, so those are our different types of neuronal synapses. Now, I want to talk to you a little bit about the different types of synapses that we can get. So we have electrical synapses and we have chemical synapses. Chemical synapses are way more common. So I'm not going to spend a ton of time talking about this kind, electrical. I'll talk about it briefly. An electrical synapse is basically when you have neurons that are coupled together, okay, with gap junctions. So think of the term gap junction. A gap junction is there's just a hole. So it's sort of a protein that's holding these neurons together, but there's a hole between them, and it's allowing molecules to move back and forth between the two neurons that are coupled together. And if it's allowing these ions to move between the neurons, that means that the neurons are coupled together. So if one neuron is sending an action potential, which is an electrical signal, then the neuron it's coupled with is going to be sending an action potential, right? So this is why we call this an electrical synapse, okay? This is super fast. So the transmission is very fast between these neurons when it's an electrical synapse. And we also say that when you've got neurons that are coupled together this way, they can send action potentials or impulses simultaneously. So they're sending these impulses very quickly at the same time, which is really important because it allows your neurons to be synchronous. It allows them to send signals together in unison. This is important for a lot of the functions that are going on in our brain. So we need this for things like arousal from sleep, when you need neurons firing together to wake you up, or to pay attention, right? We need electrical signals for that. Even your emotions and your memory, and even maintaining homeostasis like water balance, all of these are electrical synapses, okay? Now, chemical synapses, I mentioned, are way more common. These are the ones that you already know of, right? These are the ones that we talked about in muscle physiology, where you've got your presynaptic neuron coming in, you've got this synaptic cleft in here, okay? And then you've got this postsynaptic neuron down here, so it's got to jump the gap. And so we are releasing, remember, a neurotransmitter, which is a chemical. So that's a chemical synapse, way more common. And so these are releasing, remember, neurotransmitters. Now, the neurotransmitter that you are already familiar with is acetylcholine. That's the one that we talked about in muscle phys. But we have, honestly, we've identified over 50 different neurotransmitters. And so we always find within our chemical synapses, we always find two components. There's an axonal terminal, okay? The axonal terminal, I always draw it, you can see it, it's up here. I always draw it as sort of a bulge, right? So it's sort of that bulge at the end of the axon where you're storing those synaptic vesicles with the neurotransmitter inside. And then there's always going to be a receptor region. This has to be on the postsynaptic neuron, right? In muscle phys, this is our receptor region. In neurophys, that's our receptor region. It's the same thing, okay? It's got to have receptors that will pick up that neurotransmitter that's being released. The synaptic cleft, y'all are already familiar with this. This is that fluid-filled space that separates the presynaptic and postsynaptic neuron. And it's really important because having that space between the two makes sure that the impulse can only move in one direction, right? I mentioned this in muscle phys. If you have this space where you're releasing a neurotransmitter from the presynaptic neuron and you're picking it up on the postsynaptic neuron, the only way that impulse can move is this direction, right? It can't possibly go backwards. That's impossible. And it's impossible for a couple of reasons. One, the postsynaptic neuron down here does not have any synaptic vesicles in it, right? We don't have synaptic vesicles down here. We can't release a neurotransmitter from here. And the presynaptic neuron up here doesn't have receptors to pick it up. So we can't possibly send a signal in the other direction. So this ensures that impulses are going in one direction only, okay? And this is a chemical event, right? It requires a neurotransmitter. And so in terms of the chemical synapse and what's happening at the chemical synapse, you should already be familiar with this because this is exactly what we did in muscle phys. So you've got the words right here. I am just going to pull up a whiteboard and draw it out. Okay, so you have your presynaptic neuron. Okay, in this case, this is our postsynaptic neuron down here. Okay, remember, we've got our receptors down here. We've got our synaptic vesicles that contain a neurotransmitter. Now, if we're not looking at muscle phys, the neurotransmitter can be any of the neurotransmitters we've identified. So something like not just acetylcholine, but it could be dopamine, or serotonin, or epinephrine, or any other neurotransmitter. We'll talk about different neurotransmitters in just a minute. Okay. So remember, what's happening at the synaptic cleft is an impulse comes down at presynaptic neuron. The next thing that's going to happen is calcium channels will open. That influx of calcium causes these synaptic vesicles that contain the neurotransmitter to migrate towards the end of the axon. And then they release that neurotransmitter into the cleft. The neurotransmitter diffuses across the cleft from high to low concentration and binds to receptors on that postsynaptic neuron. That is going to cause sodium channels to open, and that is going to depolarize that membrane and lead to an action potential moving down the postsynaptic neuron. Okay, so this should look identical to muscle phys. Once we've gotten that action potential moving down the postsynaptic neuron, we've got to get rid of the neurotransmitter in this cleft. Now, we know how we do that with acetylcholine. We use acetylcholine esterase. It's an enzyme that will specifically break down acetylcholine. How we do that with other neurotransmitters, it just depends on the neurotransmitter. Some neurotransmitters will get broken down by enzymes like acetylcholine. Some neurotransmitters actually will get sucked back up. They'll get vacuumed back up into this presynaptic neuron up here. Okay, so it really depends on which neurotransmitter we're looking at as to how we get rid of it. Okay, so let's go back to that PowerPoint. Right, so we just walked through all these steps, right? So the impulse gets to the end of the axon, calcium channels open. That causes the neurotransmitter to get released into the cleft. Okay. That neurotransmitter is going to diffuse across the cleft and bind to those receptors. And that's going to cause the membrane potential to change, right? Sodium channels open, the membrane potential is going to change. Now, one thing I will tell you, and we're going to talk more about this in just a few minutes, is that when sodium channels open, they can either make that membrane potential excited, which will lead to an action potential, or they can even inhibit the membrane potential, which means you won't get an action potential. So some neurotransmitters are excitatory and some are inhibitory. And again, when we talk about neurotransmitters, I'm going to touch on this and how that works. Okay? And then lastly is once we've gotten the effect on our postsynaptic neuron, whether it's excitatory or inhibitory, we got to get rid of the neurotransmitter, right? With acetylcholine, it's acetylcholine esterase. With other enzymes or with other neurotransmitters, it might be something different. Okay? Again, this should look super familiar. This is the picture from your book showing you the same concept, right? So we've got that impulse coming down, got our calcium channels that are opening, synaptic vesicles then migrate to the end and release the neurotransmitter, binds to those receptors, and that's going to change that membrane potential, either excitatory or inhibitory. Okay. Now, obviously, when we're dealing with a synaptic cleft, we do have a delay. We call this synaptic delay. This is the time it takes for calcium channels to open, synaptic vesicles to migrate to the end and release acetylcholine. This takes time for it to bind to receptors, for sodium channels to open. And so all of this takes time. And so this is truly our rate limiting step. Now, the time that this takes at the cleft is not long. It's on the order of 0.3 to 5 milliseconds. So it's still fast, but it is the slowest part in neural transmission. Okay, everything else is incredibly much faster than this. And so this is just sort of our rate limiting step. All right, so I'm going to walk you through some of the neurotransmitters now, since we're talking about our synapses. Neurotransmitters are how your neurons are communicating with each other and with effectors, right? We already know of acetylcholine. We've identified 50 different neurotransmitters. Most neurons will release more than one type of neurotransmitter. Usually they'll release at least 2 or more neurotransmitters. And the reason for that is it allows that presynaptic neuron to have different effects on its effector. So but if it releases one neurotransmitter, it might be excitatory. If it releases a different neurotransmitter, it might have an inhibitory effect. And so it allows the cell to have different influences. And we classify our neurotransmitters chemically and functionally. Okay, I'm going to walk through the chemical classification first. This is not all of the chemical classifications. This is just some. These are the ones that I chose from your book that I thought you would probably know the most about, or at least be the most familiar with. Okay, so acetylcholine we've already talked about, but I'll touch on it again. We also have neurotransmitters that are classified as biogenic amines. Okay, we have neurotransmitters that are classified as peptides and purines. Okay, so let's go through just some of these classifications. Acetylcholine, this is the one that is released at the neuromuscular junction. And this one was actually the very first neurotransmitter that was identified. And so because of that, we've studied it the longest and we know the most about it. This one is made and housed in your synaptic vesicles. It is degraded by acetylcholine esterase. And it's released by any neuron that stimulates your skeletal muscles, and even by some neurons that are part of that autonomic nervous system. So we talked about that at the beginning of chapter 11. So remember, the autonomic, think automatic. So this is the part of your nervous system that is regulating those things that you have no control over. So things like sweat glands, oil glands, your heart rate. This is your autonomic nervous system. You have no control over when that stuff happens, right? So acetylcholine can get released there as well. Now, a big classification of neurotransmitters, the second one, is something called biogenic amines. Now, by itself, biogenic amine might mean nothing. But these include things like catecholamines and indolamines. Catecholamines are things like dopamine, norepinephrine, epinephrine. Those should sound familiar, right? And indolamines are things like serotonin, histamine, right? Hopefully y'all are familiar with histamine. If you've ever taken Benadryl, that's an antihistamine. So histamine is the stuff that your body will release, and it causes your capillaries to get leaky, and it causes your mucous membranes to get leaky, which is why you get the watery eyes, you get the runny nose, you might get some swelling when you have histamine being released. And antihistamine counteracts all of that. Other ones like dopamine and serotonin, you've probably heard of that if you've ever seen any commercial on TV for an antidepressant. Usually their buzzwords are serotonin and dopamine. So I always think of those neurotransmitters as the happy neurotransmitters, okay? So we can say that for biogenic amines, they can be excitatory or inhibitory. It just depends on the receptor type. These we find broadly throughout the brain. So we have these neurotransmitters are being released all over in the brain, but we definitely know they play a huge role in emotional behavior. Again, think serotonin, think dopamine. Now, one thing that you all should know by now in anatomy is that too much of anything is bad. So just because dopamine and serotonin are your happy neurotransmitters, too much of them is definitely not a good thing. In fact, too much of these can actually lead to mental illness. It can lead to things like schizophrenia. Just like too little is not good. Too little dopamine and serotonin can lead to depression. So you don't want too much, you don't want too little, you want it just like Goldilocks to be just right, okay? Psychoactive drugs, which we'll talk about at the end of the chapter today, like LSD, these can actually bind to the receptors that normally pick up biogenic amines, and they can cause wild hallucinations. And we'll talk about how that happens in a little bit. Now, we also have neurotransmitters that are amino acids. Now, that term should be so familiar, amino acid. Amino acid is a building block of our cells. If you hook amino acids up together, you get a protein, right? You should know that from general biology. Lots of amino acids strung together as a protein. So amino acids are incredibly common in cells, but they can also be neurotransmitters. So I'm going to diverge just for a second. One of the things that we're going to talk about, and I know I've touched on it already, one of the things we're going to talk about is something called the blood-brain barrier. We'll talk about this in the next chapter when we do the brain. If amino acids, which are the building blocks of proteins, you eat proteins, your body breaks them down, you absorb the amino acids, you have these in your bloodstream all the time. And so the last thing you want is for these chemicals that are found in your bloodstream to get into your brain, because these can act like neurotransmitters. And so this is one of the many reasons why we have a blood-brain barrier. It keeps what's in your blood away from your neurons, because a lot of these neurotransmitters that we're talking about are things that are very common in our bloodstream. And if these were able to make it through the blood-brain barrier, your neurons would not be able to function correctly. So we have to keep all that stuff out, okay? So because amino acids can act like neurotransmitters, we do have two different kinds that I'm going to mention that we find in the brain that are getting released by neurotransmitters. One is called GABA, and I'll bring this back up when we talk about drug addiction, specifically alcohol. GABA is a neurotransmitter that is typically inhibitory. Okay, so we're used to thinking about neurotransmitters as excitatory, right? Think of acetylcholine. It causes your muscle to contract. GABA does the opposite. It prevents anything from happening in the postsynaptic neuron. So it's an inhibitory neurotransmitter. This is the same neurotransmitter that is exacerbated by alcohol. So what that means is that when you drink alcohol, it actually causes the GABA, it causes it to be exacerbated. So it's even more inhibitory. So think of all the things that happen when you drink alcohol. Your speech slurs, your vision becomes impaired, motor reaction time is slowed down, everything gets slower, right? And so this is one of the reasons why. GABA is already inhibitory, alcohol makes it more inhibitory. Glutamate is another one of our amino acid neurotransmitters. And again, I'll mention this one again when we do drug addiction here at the end. This one is actually really important in learning and memory. So one of the things when we talk about drug addiction, why drugs are so addictive is that this is the best example I got from a student one semester who was an AA and an NA counselor, a sponsor. And he told me, he said, you know, when you get a high for the first time, it would be like the size of a basketball. And every time after that, when you take that drug, the high gets smaller and smaller and smaller. And you're always chasing the basketball. You're always chasing that first time you got high, and it just felt so good. And so glutamate is the neurotransmitter that helps you remember what it felt like when you got that high the size of a basketball. And so glutamate is important in learning and memory, but it is also one of the reasons why drugs are so addictive, is because you remember that feeling. We also say that glutamate is the stroke neurotransmitter. So what happens when somebody has a stroke? Strokes are usually caused by a blockage of blood flow to your brain. So it could be from a blood clot that gets stuck. It could be from maybe a mass or a tumor that's growing and pushing on the blood supply. But when someone has a stroke, if they cannot get oxygen and nutrients to nervous tissue, the nervous tissue will start to die off. And one of the reasons why this has been dubbed the stroke neurotransmitter is that when you block oxygen and nutrients to your neurons, it actually causes those neurons to release a huge amount of glutamate. They just pump out tons of glutamate. Again, too much of anything is always bad. And all that glutamate in the synaptic cleft leads to excitotoxicity. It's exactly what it sounds like. You overexcite your neurons to the point of death. It's toxic to them. Okay? So glutamate is important in learning and memory, but it's also the stroke neurotransmitter. Again, and this is because when someone has a stroke, those neurons die quickly without oxygen and nutrients, but also they're dying because of all that glutamate being squeezed out. So most people, when they have a stroke, they do experience some kind of deficit that comes along with that. Now, I think this is one of the last ones. We also have neurotransmitters that are peptides. Again, polypeptide. That should be a term you've heard before. A polypeptide is a protein, right? So peptides are just strings of amino acids. Endorphins. You know what endorphins are. These are natural opiates that help you inhibit pain. I'm not a runner. I'm too old for that. I never really enjoyed running anyway. But I had a friend in grad school who used to run, and he would run marathons, and he would run miles every day. And he said that he had a runner's high. Maybe you've heard of a runner's high before. It's when you run for a really long time and eventually you start pumping out tons of endorphins and you feel high. You feel great and it hooks you to the sport apparently forever. You're not getting a runner's high every day, right? But you are releasing some endorphins when you engage in behaviors that are good for you or good for the species, right? So even when you work out, just going to the gym and working out, nobody ever wants to really go to the gym. You always go and you kind of drag yourself in. And then when you leave, I don't think anyone has ever left the gym saying, man, wish I wouldn't have done that. You always leave feeling better. You feel happier. Because even just working out, doing exercise, actually causes you to release endorphins. Not to the point of a runner's high, but it causes you to feel good. You've done something good for your body and you are getting rewarded for that. Now we can mimic natural endorphins with drugs. So drugs like morphine, heroin, methamphetamine, these are all drugs that mimic our own natural endorphins, and they are incredibly addictive. I used to work in the hospital. So when I graduated from Ohio State University, I got a job working for an intraoperative monitoring company. So I worked in the hospital. I worked with neurosurgeons. My job was to monitor the integrity of a patient's nerves, peripheral nerves, and central nervous system throughout the surgery. So usually the surgeries were things like slip discs, herniated discs, scoliosis, brain tumors, anything involving the brain and spinal cord. And a lot of the neurosurgeons during surgery, they would chat, we would have conversations. And one of the surgeons had his prescription pad stolen that week. So a lot of these neurosurgeons would do like they would see patients in the office like on Monday and Tuesday, and then Wednesday, Thursday, Friday, they would do surgeries, okay? And so this doctor had had his prescription pad stolen earlier in the week. And he found out because the pharmacist called him. So a pharmacist called him and said, hey, Dr. Smith, I think your prescription pad has been stolen. I just got a prescription written with your name for it was supposed to be morphine, but the patient really misspelled morphine badly. So the pharmacist knew that this was definitely not the neurosurgeon writing this prescription. But that goes to show you how incredibly addictive these substances are. You know, this obviously you go to jail for stealing a prescription pad and forging a prescription. This is the last one, I think. So this is purines. So again, this is another one of those terms you probably have heard of. You may have to brush off the cobwebs. So purine and pyrimidine, maybe that sounds familiar. So if y'all remember DNA has base pairs. It has A's and T's and G's and C's. And we classify those as either purines or pyrimidines, right? And so adenosine, adenosine is our A, right? This is one of the bases in DNA. So it is a purine. And this also, not only is it a base pair for DNA, but it can act like a neurotransmitter. So adenosine is normally an inhibitor in the brain. So when it gets released into that cleft, it's not causing an action potential. It is stopping action potentials. But caffeine will actually block adenosine receptors. So let's draw this out so you know what I'm talking about here. Okay, so let's say here in our synaptic cleft, we have some adenosine here. And we have receptors that normally pick up adenosine. And adenosine will block any action potential from moving down this postsynaptic neuron. That's its normal job. But when you drink caffeine, so Mountain Dew, coffee, whatever it is, that caffeine can move across the blood-brain barrier. It can get into your brain, and it can bind to these receptors and cause an action potential. Well, if you've got caffeine on the receptors, guess what can't attach to the receptor? Adenosine. So you're going to be getting an action potential when you shouldn't be. So this is why when you drink caffeine, you get jittery, right? You get jittery, your heart races, you get all those things that come with drinking caffeine. I had a student tell me once that he drinks 2 liters of Mountain Dew every day. I had him for AP1 and AP2, and I think by the time we ended in AP2, he was down to like 1 2-liter of Mountain Dew a day. So we worked on it over the course of 2 semesters, getting him to sort of wean off that Mountain Dew. So I told you that we've got our chemical classification of neurotransmitters, which is what we walked through, but we also have our functional classification. So this is that neurotransmitters can be excitatory, but they can also be inhibitory. Excitatory neurotransmitters, these will cause depolarizations. They lead to an action potential, okay? So examples would be like acetylcholine or glutamate. These are excitatory neurotransmitters. Inhibitory neurotransmitters, they cause hyperpolarization. So let's go back to, I'm going to pull up a whiteboard. So let's clear this, okay? And let's go through, oops, that's not what I wanted to draw. Okay, let's go through this drawing that we did earlier in chapter 11, okay, where we start with resting membrane potential. Remember, we're sitting at -70 millivolts. Inside's negative, Outside's positive, more sodium out, more potassium in, okay? Depolarization, this is when sodium channels open. Sodium comes in because it has a positive charge. It makes the inside more positive. The outside becomes more negative. If we get positive enough, we get to threshold, which is negative 50 to 55 millivolts. Okay? And that will lead to an action potential. Right? And an action potential means we got lots of these sodium channels opening, and the inside is getting up to positive 30. Okay? After we get that action potential, we've got to get back to resting membrane. So we do that by repolarizing our membrane. So remember, we're going to close our sodium channels. We want no more of that coming in. We're going to open potassium channels. Potassium has a positive charge. The outside becomes positive. The inside becomes negative. We get back to negative 70. But in the process of repolarizing, remember in neurophys, sometimes we leave these potassium channels open a little too long and we overshoot. And the outside becomes too positive and the inside becomes too negative. And we call that hyperpolarization. Okay? And I mentioned to you how we have these refractory periods, which means you cannot get another action potential during this time. Our absolute refractory period is from here to here. You will not get another contraction or you will not get another action potential. Can't happen because you're already getting one. But here during hyperpolarization, if y'all remember, we call this the relative refractory period. Meaning you're probably not going to get another action potential, but you could. So the reason why I'm drawing this out for you again is because when we talk about excitatory versus inhibitory neurotransmitters, an excitatory neurotransmitter is one that causes this to happen. It opens your sodium channels. Open sodium channels. It can cause you to hit threshold, you get an action potential. That's excitatory. An inhibitory neurotransmitter is doing this. It's going to open not sodium channels, it's going to open potassium channels. So we're not going to go through all of this. Instead, when an inhibitory neurotransmitter is released, it immediately opens potassium channels. And it causes you to hyperpolarize your membrane, which means you are now in this relative refractory period and you're not going to get an action potential. Okay? So the way excitatory and inhibitory neurotransmitters work is they both change the membrane potential. An excitatory neurotransmitter is up here. It's opening sodium channels. It can cause you to get an action potential. An inhibitory neurotransmitter is opening potassium channels. That's what it opens. And it causes your membrane to drop to negative 90, which throws you into this refractory period. You're not going to get an action potential. Okay? And so this is why we say that inhibitory neurotransmitters hyperpolarize the membrane and they prevent an action potential. A good example of an inhibitory neurotransmitter is GABA. So remember, this is one that I told you it's normally inhibitory, so it prevents action potentials anyway. And it's exacerbated by alcohol, makes it more inhibitory. Okay? Now, in terms of this functional classification, it can get sometimes a little complicated because I just told you they're either excitatory or inhibitory. Well, sometimes they can actually be both. What determines whether the neurotransmitter is excitatory or inhibitory is not the neurotransmitter. It's actually the receptor that's picking it up. So it's determined by the receptor at the postsynaptic neuron, whether that neurotransmitter is excitatory or inhibitory. So a good example of this is acetylcholine, right? Acetylcholine at the neuromuscular junction. That's excitatory, right? Think back to muscle fibs, right? Acetylcholine gets released. It binds to these receptors. Sodium channels open, right? Depolarize that membrane. That's excitatory. You can get an action potential. You can get a muscle contraction. But that same neurotransmitter, acetylcholine, is inhibitory in cardiac muscle. So in cardiac muscle, it's doing this. When acetylcholine gets released to the cleft, it gets picked up by the receptors in your cardiac muscle, and it causes potassium channels to open. And it causes that inside to get more negative. It hyperpolarizes it. So it's inhibitory in your cardiac muscle, okay? So same neurotransmitter is having different effects. And again, it's dependent on the receptor picking it up. So the very last couple of things I'm going to walk you through in this chapter is just a little bit on drug addiction. So in terms of drug addiction, our brain will always give us rewards if we engage in any behavior that is good for us or good for our species, right? So again, the example I gave you earlier is when you work out. You work out, you release some natural endorphins. It makes you feel good. Your body is giving you a positive reward for that. How this works is dopamine, it's our happy neurotransmitter, actually gets released in the pleasure center of our brain. And we'll talk about that pleasure center of our brain in chapter 12. But this pleasure center can artificially be stimulated by drugs. Okay? So let's talk about a couple of different drugs. The first one I'll mention is methamphetamine. Methamphetamine is a neurotransmitter-like chemical. So that means that it acts like a neurotransmitter. It will cross the blood-brain barrier and act like a neurotransmitter when it gets into those clefts. The problem, and it's not just with methamphetamine, it's with lots of different drugs. The problem is that when you take a drug that mimics neurotransmitters, it causes your brain to make less of its own neurotransmitter. So another way you can think of it is it causes your brain to run dry. So when you take methamphetamine, which is acting like a neurotransmitter, your brain says, oh gosh, I got plenty of that neurotransmitter. I don't need to make more. And it stops making it. You know, I had mentioned my student who is an NA and an AA counselor, and he said that he recently had someone he was sponsoring call him. He had been sponsoring this young man for 8 months. 8 months. The young man called him. He had been clean for 8 months. Called him so happy and excited because he had his first genuine laugh since being clean. Think about that. That's because your brain is no longer making some of these happy, feel-good neurotransmitters. It's dry. It takes that long for your brain to catch up and start making these neurotransmitters again. So I know that sounds weird, but to have a genuine laugh, we take that for granted. Well, someone who is a drug addict, they're not going to have a genuine laugh for a long time, even after being clean. And this is why, another reason why trying to get clean is so hard for so many people. Another drug that I'll mention is cocaine. Cocaine is a drug, does not act like a neurotransmitter. Instead, it actually blocks the reuptake of a neurotransmitter. So remember how we talked about like acetylcholine after it's done its job, we have to get rid of it. We use acetylcholinesterase to get rid of it, so it's not hanging out in the cleft. Dopamine, when dopamine gets released, how we get rid of it is we suck it back up. Okay? We have a reuptake transporter, sucks it back out of the cleft, so it stops the action. Cocaine works by blocking the reuptake of dopamine. So what happens is dopamine stays in that synaptic cleft, and you keep getting an impulse in that postsynaptic neuron over and over and over again. And this is that high feeling. You get that long-lasting high when you take cocaine. Again, though, what's going to happen is your brain's going to run dry, because now you've got all this dopamine in your cleft. Cocaine is preventing it from getting taken away. And if it's hanging out in the cleft, your neurons are going, gosh, we got a lot of dopamine. We don't need to make any more. And it stops making dopamine. And remember, dopamine, it's one of our happy neurotransmitters. So this is going to lead to that addiction cycle. Cocaine is needed to experience pleasure, but the more cocaine you take, the less dopamine your body is making. You don't have very much. And so it causes people, this is why people end up overdosing. Because again, they're chasing the basketball. And so they're going to take more and more and more of that drug to try to get that big high, but they can't get that big high anymore because they're not making as much dopamine anymore. I mentioned glutamate. Remember, this is our learning and memory neurotransmitter. This is the one that's responsible for helping us remember what the basketball high felt like, right? And this one plays a huge role in drug addiction. Now, for cocaine, there are some drugs that can be given to people who are addicted. I don't like this word. This is the word that your book uses. I don't like the word vaccine here. This isn't a vaccine. I think of a vaccine as something that prevents illness, like it's going to prevent you from getting smallpox or whatever it is. But I think of this as more of, these are medications that you can take. So there's a drug that will prevent cocaine from ever reaching the brain. So it basically prevents cocaine from passing through the blood-brain barrier. And these are all drugs that are in various forms of clinical trials. There is one that is on the market. It's called vanoxarin. This is one that helps to even out the highs and lows. So one of the issues with someone who is addicted to cocaine is that when you take the drug, it blocks the reuptake of dopamine. So dopamine hangs out in the cleft. But as soon as the effects of cocaine wear off, guess what happens to all that dopamine? It gets sucked up instantly. So you go from a really high high to a really low low, and you crash. And again, this is one of the reasons why it's so hard to get clean. And so vanoxarin is a drug that blocks the reuptake transporter, and it helps to steady the reuptake of dopamine. So it takes it up slower so that you don't crash as hard. It prevents that crash. And then there's also a drug that will help to prevent sort of the cravings that go along with drug addiction. I had another student a couple of years ago who, it is a really sad story. She's very smart. She had a boyfriend who was addicted to pills, and he used to have pill parties. And he would always ask her, oh, try this or take this or do this. And she was always like, no, I'm not doing that. And one day she said she just had a really bad day. And he asked her the worst time possible. And she said, okay. And she took some prescription pain pills, and that was all it took. She became addicted. She said she could not function without 8 to 10 Loratabs a day. I've never had Loratab. My husband had foot surgery and was on Loratab for like a week. And he said, like, it's dangerous stuff. And he's like, how could you function on that many Loratabs a day? He's like, I would take one after surgery and be out for like 2 days. And so that's what happens. People get so addicted that they can't function without it. And so she was a student of mine. She was still taking prescription pain pills, but she was trying to get clean, and she was taking Suboxone. So Suboxone is a drug that people take when they are addicted to things like methamphetamine, prescription pain pills, narcotics. And you're supposed to, when you take Suboxone, not take anything else. And you are supposed to start at a dose that prevents you from having any kind of withdrawals and gradually wean yourself off of it under a doctor's care. And she said that she had not weaned down in a couple of months because every time she tries, she has some type of withdrawal, and she wanted to experience no withdrawals. And I told her that's probably not realistic. If you're addicted to drugs and you're trying to get clean, you're probably going to have some effects of withdrawal. And so, no matter which of these drugs you're taking, if you're trying to get yourself clean, you're going to feel it. It's not going to feel good. We know that addiction is strong. I had an aunt who smoked cigarettes and drank. And those are both legal things. And we know how addictive those are, right? She ended up getting lung cancer and had not a lobe removed, but the entire lung, her entire left lung was removed. So she only had one lung. And they told her at the time, you have got to stop smoking. You're going to die. When they removed her lung, they gave her 6 months to live. She lived for another 12 years, smoked every day. She could not give it up. She would smoke with oxygen, the oxygen cannulas in her nose, and she would still smoke cigarettes. And we know oxygen is highly flammable. And so we know how addictive drugs can be, even the legal ones. And unfortunately, though, it results in people doing very odd things. They do these weird combinations. Meth labs. Meth labs are this is just how people make meth. It involves pseudoephedrine, so it involves that cold medicine. This is why you have to show your license when you buy pseudoephedrine. And if you buy too much, they'll deny you from getting any more. My son plays hockey, and last year we flew to Maryland for a hockey tournament, and I got sick. I had a cold, like a head cold. And when you fly and you have all the gook and the yuck, it hurts your ears so bad. And so I had to go in and buy some Sudafed because that'll dry you up so great. But because I talk about this, I'm always paranoid buying Sudafed. And I'm out of state. I have an out-of-state license. And so I go up to the counter and I'm buying Sudafed. I'm explaining my whole life story to this lady and why I'm buying Sudafed. And my husband's like, shut up. Nobody cares. And so, it makes I'm super honest. So it makes me feel like I've got to tell my whole life story so people don't think I'm making meth in a trailer somewhere. But cold medicine, match heads, iodine, and acetone. And these all get cooked together. Acetone is incredibly flammable. And so this is how people blow themselves up when they're trying to make meth. I'm from Cincinnati, Ohio, and meth is rampant where I'm from. It's a terrible, terrible addiction where I'm from. And every day on the news, there's something else, some other story about it. And a couple of years ago, there was a story about a couple who were making meth out of their home, and they had a toddler. And when people are making meth and they're addicted to drugs, they're not worried about anything else. Kids that are growing up in situations like this, they fend for themselves. And this toddler, the parents were both passed out, and the toddler came in to get something to drink and picked up a cup, a little purple cup, and took a drink, and it was filled with lye, which is a strong pH solution used to make meth, and the child died. The couple put the child in the car, didn't want to take the child to the emergency room because they were afraid that they would get in trouble. And so they drove around with the child in the car until the child died. It is awful. These are the kind of stories that my mom tells me about all the time from where I'm from. These are some different results and combinations that I'm not familiar with. They're from your book. Some of them are funny to me, but some of them are disgusting. So illy, never heard of this. This is marijuana that has been dipped in formaldehyde. Now, I don't do drugs, but I can tell you that I know what formaldehyde smells like, and I don't like to even just smell it. So I can't imagine smoking that. That's gross. H-bomb is ecstasy laced with heroin. And A-bomb is marijuana with heroin or opium. Sextasy, which I think is funny, not because they use it, I just think it's funny, is ecstasy and Viagra combined together. And then octane is PCP laced with gas. So again, you know, all of these weird combinations, this is the result of people chasing the basketball, right? They're trying to get that big high. So they're doing more and more. They're combining things together so that they can get that big high. But again, it's never going to happen because your brain is running dry. It's not making these neurotransmitters as much anymore. Anytime you take a drug that acts like a neurotransmitter, it is going to destroy neurons. It is going to cause a loss of verbal and spatial memory. It leads to your sleep-wake cycle being destroyed. So when it's time to go to bed, when it's time to wake up, so sleeplessness is really common. Depression is really common. Memory problems. And one thing that's important to remember is that your brain will always try to circumvent any euphoric state that you put it in. So you will always come down from the high, always.